is tell you about our more recent work on the development of a toolbox for fluorine magic angle spinning and dynamic nuclear polarization. And I'll talk about quite a few types of systems, starting with small molecules and pharmaceutical formulations, going to protein assemblies, microcrystalline proteins, and if I have time, also protein-ligand complexes. And so specifically, we'll talk about considerations for magic angle spinning frequencies. You will see that spinning faster is invariably better. We'll talk about how to measure fluorine chemical shift tensors, how to measure accurate interfluorine distances in multi-spin systems, how to structurally characterize active pharmaceutical ingredients in blockbuster drugs, 2D and 3-dimensional fluorine-based correlation experiments to look at resonance assignments and also determine long-range distance restraints, and our results on dynamic nuclear polarization fluorine-enhanced NMR. So this in a way continues the topic that Andy started this morning, a weird nuclei in biological systems. Okay, so, and as most of you know, fluorine is not commonly found in biological systems with very few exceptions. Okay, so, but it's a very nice NMR probe. Actually, those of you who are not aware, fluorine has the second highest of all stable isotopes gyromagnetic ratio after proton. Okay, so it's a spin 1/2 nucleus. While it's absent from naturally occurring biomolecules, it can be actually readily incorporated into proteins and nucleic acids without causing major structural perturbations. Another thing which is important is that about 30% of all pharmaceuticals on the market currently contain a fluorine. And because of high gyromagnetic ratio of fluorine, distances between fluorine atoms of the order of 20 angstroms are readily available from the measurements. Right, so if you look at the literature, and I should say that by no means are we the first to look at fluorine NMR in biological systems, there is very nice literature going back to 1990s at least, but if you look at what people have done prior to several years ago, you will see that most of the magic angle spinning experiments were performed at frequencies of 20 kilohertz or below. So one of the first questions we asked ourselves for several years ago, and actually without having proper hardware at hand, we used the Bruker HCN probe tuned to a fluorine channel without decoupling. Anyways, we had 2 fluorotryptophan samples shown here, and we have done just one-dimensional experiments going from 20 to 60 kilohertz spinning frequencies. And not surprisingly, what you see here is that spinning faster gives rise to higher resolution and higher sensitivity. More recently, we were able to, in collaboration with Joachim Stulpe from Bruker, go up to 111 kilohertz. And these are spectra for atorvastatin calcium, also known as Lipitor. And you can see that going to even higher frequencies actually results in better sensitivity and resolution. Decoupling also helps. You will see that even at 100 kilohertz, you do need proton decoupling. Right, so one thing that is very useful is to know chemical shift anisotropy tensors and fluorines. If you look at the literature, actually there are not many reports on CSA tensors, and most of the measurements were done at slow spinning frequencies. And indeed, using this antimalarial drug mefloquine spinning at 10 kilohertz, you can record the spinning sideband patterns, but extracting CSA from that is prone to errors. So we decided, Galpor, Dallerbrook, in the lab in collaboration with Joachim Stulpe, implemented the RN symmetry-based sequences, which were first developed by Malcolm Levitt, and adapted them to fluorine measurements. We've done some RN-CSA measurements in our lab on all kinds of nuclei and proteins, and you can see here that for mefloquine spinning at 60 or 100 kilohertz, you can recouple the line shapes very nicely. One thing to point out is that even spinning at 100 kilohertz MAS with 2 combinations of symmetry numbers, you can get nicely recoupled line shapes, but this is what happens without decoupling. Okay, so you see substantial broadening, and as the result, a lot of errors in the derivation of the CSA tensors. Right, so now, what kind of other structural information can we extract from fluorine-19? One of the questions we asked ourselves is whether we can measure intrafluorine distances in multi-spin systems. Again, going back to the classical literature, you will see that measurements in the past were done by isotopically diluting the fluorine-containing molecules to avoid multi-spin effects. Okay, so but obviously this is not ideal. If we're working with pharmaceutical formulations of proteins, you have multiple fluorine sites, so you should be able to record distances in these multi-spin systems. Can we do that though? To answer this question, we turned to a number of model systems. These are difluorobenzoic acids containing fluorine substitutions in different positions. And you see here that for each of the two fluorine atoms, we have distinct peaks, which we were able to assign. The chemical shifts span quite a nice range. Also 7-ethyl-tryptophan, which has one fluorine, obviously, but it has two peaks because of the two crystallographically inequivalent sites in the molecule. Right, so how do we measure intrafluorine distances? We do a classical now RFDR experiment, and one of the first groups to do that was back in 2001 and McDermott's lab. So we did this experiment in two flavors, one using just a two-dimensional experiment and recorded spectra as a function of RFDR mixing time. Another way to do it is to use selective Dante inversion pulses and follow that by the RFDR mixing. And you can see the exchange and build-up curves here. Now, this is 2,4-difluorobenzoic acid. What happens if we try to simulate those curves, just assuming a single spin pair intrafluorine distances, which are intramolecular? This is what happens. It looks horrible, obviously, right? Not a surprise. Now, if we do a multi-spin system simulation, taking into account this three sets of distances, intra and intermolecular, you can see that the agreement between the experiment and the simulations is quite nice. Right, that's nice. What about other examples? Let's look at 2,5-difluorobenzoic acid, a slightly more complicated case. Again, single spin pair simulation fails. But if we do multi-spin system simulation, and here you have to also account for the effect of the positional isomerism and the relative occupancies of those positional isomers known from the crystal structure, you can see that there is a nice agreement. All right, great, but you're gonna say that you guys know the crystal structures for each of those compounds, so what good does it do? Can we do this in the absence of the a priori knowledge of the crystal structures? And to answer this question, Matt Fritz, a student in the lab back then, constructed a grid of 3,005 spin systems where he actually took representative examples from the crystal structures to account for the different distances between two neighboring spins and three spectator spins. And he calculated the exchange curves. And when he did the best fit to the experimental data, you can see that the fit is quite reasonable. Actually, the distances from the fit matched very nicely to the X-ray structure. Right, so what does it mean? It means that even in the absence of the a priori crystal structure information, you can at least get an idea of what kinds of distances contribute to the exchange curves. And important to point out is that if you're working with small molecules, it's usually enough to have 5 spin system simulations and not go to larger systems. Right, so now these were all model compounds. Can we get information on real systems? So to be able to do that, we had to go and ask a bunch of our friends for blockbuster drugs that they were, willing to share with us. And this was all during the COVID pandemic. So we had a little bit of fun with this project, okay? So, and we're able to get 7 types of formulations of these blockbuster drugs, all right? So Celebrex, which is an anti-inflammatory, Prozac, antidepressant, Crestor and Lipitor, these are 2 cholesterol-lowering statins, and 3 antibiotics, Cipro, Levaquin, and Zyvox. Right, let's look at what these are. All of them have fluorine, different functionalities, fluoromethyl groups, and then single substitutions in the different types of aromatic rings here. Another thing to point out is that the fraction of the active pharmaceutical ingredient, which is the fluorinated molecule, varies widely across these compounds from 6% and some of those all the way to 79%, right? And also for 3 of those compounds of 3 of those pharmaceuticals, we had compounds from different companies, different formulations, right? So let's have a look at the fluorine spectra here, and here they are. And you can see some quite nice surprises. So first of all, the trifluoromethyl group containing compounds Celebrex and Prozac give very sharp lines. Okay, so crystalline environment dynamics because of the trifluoromethyl group rotation. In contrast, rosuvastatin in all 3 formulations gave this really awful looking spectrum, indicating that this is an amorphous formulation. And the rest of the compounds, 4 of them gave also relatively sharp spectrum, meaning that they're in crystalline environments, but there are some surprises. Look, single fluorine in the molecule, we have a bunch of peaks. I'll come back to that in a second. Right, so before I do that, let me first mention a few words about the sensitivity. We're doing these experiments in small rotors, 0.7 and 1.3 millimeter rotors. And we're working with tiny amounts of fluorinated API ingredients for the compounds where the fraction of the API is very low. So 1.3 millimeter rotors, 0.2 milligrams, as low as 0.2 milligrams of the formulated of the fluorinated API. Despite that, we can actually see strong fluorine signals in a single scan already, right? So, and we collect high quality fluorine spectra only in a few minutes, right? So if you have a long relaxation time in some of those formulations, it's as long as 10-15 minutes, but also I should say that this spectra report directly on the fluorinated API, not on the excipient. Right? So in contrast, if you want to collect carbon-13 detected spectra, you need to spend quite a few hours of signal averaging. And most of the information you get is about the excipient as shown here. Complex spectra, not very interesting. All right, great. We can detect fluorine with high sensitivity in these pharmaceuticals. What information can we get about the structure? Let's look first at one example. It's levofloxacin. And the work was done by Roman Zadorozhny, who's sitting here in the audience. So I'll talk about what he's done. And so if we collect a fluorine-19 spectrum, you can see that despite the fact that there is a single fluorine atom, you see 4 peaks, or 2 sets of 2 peaks to be more precise. And the intensities between each of the 2 sets are the same. If we do fluorine-fluorine RFDR experiment, you can see that, again, these are 2 separate sets of peaks with cross peaks in between. What does it mean? Well, right away, just from this information alone, you can tell that A, there are 2 polymorphs in both formulations, which are not cross-talking, and 2, that the proportions are different between the different manufacturers, but the polymorphs are the same. Right, great. What else can you tell? Well, to be able to do that, we go again and record the Dante RFDR exchange curves. And you can see that the exchange curve between peaks 1 and 2 is dominated by a short distance, while the exchange curve between peaks 3 and 4 is dominated by some much longer distance. Is that all we can tell? Well, turns out no. Roma went ahead and looked at the Cambridge crystallographic database, and he identified 2 forms of the compound, which actually form on the left is consistent with the polymorphs, polymorph shown in peaks 1 and 2. Right, so and then we also did quite a few more fluorine-based experiments from which you can get resonance assignments. Right, so another example is atorvastatin. Okay, so atorvastatin calcium, we had formulations from 2 manufacturers, Teva and Apotex. I should also say that, of course, these are proprietary, so we have no idea what's in them, right? So, but we can guess based on these experiments. And you can see here that, again, there are 2 peaks, or in the case of Teva, there are cleanly 2 peaks. In the case of Apotex, they also have some fine structure. Right, so the exchange curves, RFDR exchange curves, are consistent with longer distances. Roma and Kate collected some other experiments, and they were able to identify on the basis of the chemical shifts and also this exchange curves that sample 1 from Teva is consistent with what's called form 1 in the Cambridge database, which is lamellar packing type of structure. And there are 2 different environments and 12 angstrom distances in between. Right, so what I hope I've shown you so far is that you can get very nice information from fluorine-based experiments on pharmaceutical formulations. These experiments are not only very sensitive, but they're also very specific. Right, so what about proteins? Can we do fluorine NMR in proteins? We need to know how to incorporate fluorines into proteins, and there are several ways of doing that. Today, I will only tell you how to work with fluorinated tryptophan residues in proteins. And it's very easy to do the incorporation following the protocol from Peter Crowley developed back in 2012. To put fluorotryptophans, you can use fluoroindole precursors, and he's developed it for 5-fluorotryptophan. You can do it also for 4, 6, and 7-fluoroindole precursors. You get high yields, and you can also introduce carbon and nitrogen isotopes as well. All right, so to see what we get, we started this work using a system that my lab has studied for over a decade now, the HIV-1 capsid protein assemblies. Capsid protein, CA protein, I will not talk about the biology or the structure, but just tell you that it's a 231 amino acid residue protein. It has 2 domains, which are alpha helical, and a total of 5 tryptophan residues, which 4 of them are in the N-terminal domain, and 1 is in the C-terminal domain. And we wanted to look at whether we can get long-range fluorine distances to help us with the structure calculation of tubes formed by this capsid protein, right? So these kinds of tubes are commonly studied by NMR and cryo-electron microscopy. They have curvature here, and one of the interesting features is that there is a critical interaction in this tryptophan 184, which is responsible for formation of the intrahexamer interface. Right, so how do the spectra look like if you just take a solution, experiment fluorine-19 in Jobion and our collaborator Angela Groninmore's lab, collected the spectra, and you can see that they're quite informative actually. You see all of the tryptophans resolved, and there is quite a bit of structural heterogeneity. Now let's look at the tubes. This is a spectrum taken without proton decoupling, spinning at 40 kilohertz, using a probe which is not designed for fluorine, okay? So, but despite that fact, you can get very nicely resolved peaks, and actually the chemical shifts agree quite well with the solution shifts, except that of course there's structural perturbations in the local environment because we're working with tubes. And as expected, tryptophan 184, which is this broad signal here, exhibits conformational heterogeneity. Now, one question you can ask is, how do you assign those tryptophans, right? And early on, what we had to do is resort to mutagenesis. So what you do is mutate one residue one by one, right? And when we didn't have proper probes, that's what we had to do to assign the resonances. I will not spend much time on it, but just tell you that now we don't have to do that. With the availability from triple resonance HFX probes from actually not only Bruker, but Phoenix NMR as well, and other manufacturers, we can get very nice proton decoupling. And this allows us to do experiments spinning at 60 kilohertz with proton decoupling and get all kinds of fluorine-based correlations to carbons and protons. And Changmiao Gu in the lab was able to get this nice spectrum, and she's working on the assignments now. Going back to intrafluorine distances, so in these assemblies, we've also done a range of fluorine-fluorine correlation experiments, RFDR and proton-driven spin diffusion at different spinning frequencies. I will not walk you through the details of this data, but we'll just tell you that we're able to see distance correlations corresponding to distances of up to 23 angstroms, which is quite useful for structural analysis. Right, so one caveat in many of the studies on large biological assemblies, of course, is sensitivity. And when we didn't have proper hardware and could not decouple the protons, we turned to dynamic nuclear polarization to see if we're able to get any signal enhancements and do heteronuclear correlation experiments exploiting DNP. To prepare samples, we added Amupol in different concentrations, and you can see this is a spectrum I already showed you at room temperature. In the presence of Amupol, there is some broadening of the surface tryptophans, not unexpectedly, right? So we drop the temperature and then turn the gyrotron on, and we see very nice concentration-dependent signal enhancements. Okay, so now what's interesting about this fluorine-based signal enhancements is that they are of the same order of magnitude. This is this black curve here, as the proton signal enhancements are in blue. And for large Amupol concentrations, actually the fluorine enhancements appear to be slightly higher. Another thing which we were curious to understand is whether the effect is relayed or direct. And based on the build-up curves, you can see, sorry, build-up signal profiles as a function of the Amupol concentration. We know that actually the signal enhancements are the result of the direct polarization from electrons and not the relayed effect, because you can see that the build-up times for protons are threefold faster than the fluorines. Right, so now what we can do with this, of course, is get head core experiments, fluorine-carbon correlation experiments. This was done at 24 kilohertz magic angle spinning, and you can see that for different mixing times, we get spectra which look like that. I mean, I'm not really impressed about the resolution of those spectra, but keep in mind that there is no proton decoupling. And despite the fact that the resolution is not that high, Man Lu in the lab was able to assign quite a few residues here based on the comparison with the room temperature spectra. And when she mapped it on the structure of the CA protein, what turns out is that we're seeing correlations up to 12 angstroms, right? So this is a nice complement of tools. And how much more time do I have? actually almost done. Okay, so let me just very quickly tell you about an example on microcrystalline proteins, which is something which we found to be quite useful. An example on Oscillatoria gardi, this is a glutenin. It's a very interesting protein. It has this unique beta barrel-like structure, and it's built by two sequence repeats, which form a pseudo two-fold symmetry. And there is a lot of beautiful work from Angela Gronenborn's lab on solution NMR and X-ray crystallography of this protein. One thing which is challenging for studies of OA in the solid state is the fact that the sequence repeat does not allow us most of the time to resolve the resonances. So, essentially, there is a lot of degeneracy in the chemical shifts. So Brent Runge in the lab has generated a fluorotryptophan substituted OAA. He crystallized it together with the wild type OAA and together with Man Lu and Du-Won Lee from the University of Pittsburgh. They solved the X-ray structures of both. And what you see is that there is no structural perturbation upon introduction of fluorine, which to me is quite remarkable because you have 6 tryptophans here. Okay, so two of them are in the core of the protein, 2 more pairs form the carbohydrate binding loops. Okay, so with these types of samples, Brent, together with Aroma and Kate Quinn, were able to do complete resonance assignments of fluorine signals. Labels are shown here. This is based on RFDR and anti-RFDR experiments, as well as the assignments of carbon and nitrogen in chemical shifts. But what's most interesting is that now if we look at fluorine-based heteronuclear correlations, in this case, in a 3-dimensional way, at 60 kilohertz magic angle spinning, there's a proton-carbon planes from 3D experiment across each of the fluorine signals. You can see that the peaks are very well resolved, and actually there are correlations which correspond to distances up to 10 angstroms. And you can do a related experiment in a 2-dimensional fashion. The sensitivity is quite a bit higher. The resolution is as nice as in the 3D, and you can see a lot of different cross peaks. And the longest distance that Brent and Roma are able to extract from this is 16.5 angstroms. This are mapped on the structure of OA. Now, this is a model protein, but we're now using this information to calculate the structure of it. And of course, we need to use the proper force fields for fluorotryptophans, which have been just parameterized by Angela Gronenborn's lab. Right, so I'm not going to tell you much about protein ligand complexes, except to say that fluorine can also be used as a probe for interactions between ligand and the protein. This is recent work. And I will just conclude by saying that we're quite happy at the moment because we don't have a complete toolbox yet, but we're quite close. There are a few ingredients missing, but we know that we can study small molecules, proteins, protein assemblies, and protein ligand complexes using fluorine magic angle spinning NMR. And this work has been quite a few years of students and postdocs in the lab investing their intellect and time and energy into this. And I want to mention that 2 of these students are here. Roma and Sucharita in the audience. Roma is giving a poster and Sucharita is giving a talk tomorrow morning about a completely different topic. The students are Brent, Jordi Gal, Rosa, Chang Miao, my colleague Kate, who's running the NMR facility. And this is a long-standing collaboration with Angela Gronenborn and her team, and as well as people from Bruker. Without Joachim and Ivan and the late Melanie Rosé, none of this would be possible. So I want to thank you very much for listening to me and be happy to take any questions.
Thanks, Tatiana, for an outstanding presentation. So questions, please make your way to the microphones. And I'll start with one. So you put up some data for HF cross-polarization, but you didn't comment on it. How well did that work? Did that help you with the T1s?
So you're talking, it depends. The answer is it depends. In some cases, you want to do cross-polarization because proton T1s are shorter. In some cases, you don't because actually fluorine T1s are shorter. If you have trifluoromethyl groups, at least in the pharmaceutical compounds, sometimes CP is not very efficient at all. So we have examples where you actually cannot get any signal, let alone any enhancements. But it really depends on the system. And we don't yet have a systematic understanding of the prerequisites for the CP to work. Generally, it works very well. We also know that at fast magic angle spinning frequencies, we can do J-based experiments, HSQC, HMQC. I didn't show any of that here, but they work quite beautifully, actually.
That's an exciting idea, yeah. Rieko?
Then very nice talk and having different application of fluorine NMR. One of that is a structure determination you mentioned. And about that, fluorine chemical system really depends on the hydrophilicity and the solvent effect. Can you use that chemical shift information itself for structure determination?
Thank you very much for this question. And the answer is we would love to. And in fact, we've been working on trying to do accurate density functional theory calculations of fluorine shifts. And as you know, it's not that easy. And if Ilya is in the audience, he will tell you the same. So the way to do it right now is to do a thorough correlation, right? So if we have a set of model systems where we know the properties, hydrophobicity, electrostatics, etc., and to do chemical shift calculations in fluorine, you get huge errors still. So it's not that easy.
Thank you very much.
Thanks.
Hi, Tatiana. Fantastic talk. Were you expecting to see such variability in the polymorph contents from the APIs from different manufacturers? Or is that was kind of interesting to me, and I would have expected to see something that was a little more uniform. Was that surprising? Do you have any comments on that, or maybe anyone else?
Thanks for this question. I would say it's surprising and not at the same time, right? So some of these are, well, all of these are proprietary formulations, right? Some of those are generics. So a priori, we have no idea what we're looking at. So for instance, where you have two polymers of the same kind, right? So the variation is very different in the relative proportion. So I mean, I don't know if the companies came up with this independently or what happens. In other cases, in the case of atorvastatin, for example, you definitely have different polymers. So I don't know if to be surprised about it or not, but basically what we learned is that you know, you need to know what polymers you are taking as a drug, probably. And you can do those experiments, 5 minutes, you know, get your compounds, put them on the magnet, you get information right away.
Thank you for the exciting talk. I'm very interested in the HCF experiment. Can you comment on how it's done? Is it CP based or TIDO based?
So just to make sure I heard you correctly, asking about HCF
Yes.
three-dimensional experiment.
Yes.
Yeah, it's based on cross polarizations.
Okay.
So HCG and then
And the last mention is fluorine or a proton?
fluorine.
I see.
But you can, I mean, you can do it in either way, actually. You can go from fluorine to carbon to proton. It depends on the system, right? It depends on what your T1s are and in general, what information you want to extract. There's a, you know, bidirectional.
One last question.
The necessity of proton decoupling at 65 kilohertz. Is it a general statement that you don't need a proton decoupling for pharmaceutical materials and also proteins, or it depends?
So you do in most of the cases. You only don't need proton decoupling if you have some kind of other thing helping you, either dynamics or if you have a fully deuterated sample. Right, so in most of the cases we have looked at so far, even at 100 kilohertz, for instance, the mefloquine example I was showing, you do need proton decoupling if you want to measure. How to say it? You may be able to get spectrum if you want to quantify chemical shift or get, you know, higher sensitivity. You do need to decouple.
Great. Well, let's all thank Tatiana again. Okay, our next speaker is Alexandre Pulhausen, and he will, or the title of his presentation is High Field and DNP Solid State NMR to probe whole algae cells glycan composition and cell wall glycoprotein architecture.
Thank you. So, I will remove my mask. So first, it's a great honor to be here to present in front of so many people. It has been a long time, and it's actually my first ENC, but it's actually not my. Do I need to do something to change the screen? Okay, cool, perfect, thank you. So yeah, it's my first ENC, and it's such a great honor to be presenting today my PhD results, let's say. And so I'm from Isabelle Marcott's lab, but I am also co-supervised with Dora Warszawski, and I've also done some work with Professor Tuo Wang at the Louisiana State University. So my talk today will be about algae, whole cell, but also cell wall extract using solid state NMR and MAS-DNP. First, why study algae? First, they are found everywhere. They are found in freshwater, soil, oceans, but they have also many potential added values that are very interesting. First, they can be used as a nutrition for animals and humans, but also to produce biofuels. They are even now used to be therapeutics producers, but even now, some people say that it's also great candidates to be bioremediation agents, since some algae are able, for example, to biosequestrate heavy metals in water. For research, algae are also very interesting. They've already been studying quite a lot for photosynthesis questions. They are also good candidate for heterologous glycoprotein expression, which is kind of complicated in the bacteria. And finally, algae are kind of smaller or more ancient plants. So they are also interesting model for higher plant glycan. For NMR, it's also great to study, to work with, because it has a rapid growth and a well-known metabolism, making the carbon-13 and nitrogen-15 labeling quite easy to make and also not expensive because it's a photosynthetic organism. So we don't need glucose, we just need sodium bicarbonate. Finally, we already have a large library of mutants available to work with algae. And for example, here I will use some cell wall or starch mutants for this presentation. Then why study glycans of those organisms? First, because glycans are found in almost all organisms, but they are also associated with a tremendous variety of biological functions. And also compared to other biomolecules, such as protein and DNA that are linear, we can notice that glycans can be found are much more complex and, let's say, branch structure. Glycans can be found in the cell as a pure polysaccharide, and it's usually associated with energy storage, such as starch or glycogen in plants and animal cells. But it can also be found decorating other biomolecules, such as lipids, giving in algae galactolipids that are very important for photosynthesis, but can also be found decorating proteins, giving glycoproteins. And actually, protein glycosylation is one of the main post-translational modifications happening on proteins. And this glycosylation of proteins is happening through two different mechanisms, the O-glycosylation that you probably all know about, and it's involving the hydroxyproline, threonine, and serine hydroxyl group, and the anomeric carbon of glycan units. But you also have other mechanism, and the other one is called N-glycosylation, and instead it's involving the nitrogen of the side chain of asparagine, and usually associated to the anomeric carbon of N-acetylglucosamine. So first, I will talk about a methodology we developed and published last year about glycan assignment and quantification using solid state NMR on whole cell samples. So you just need to carbon-13 label your cells to have high sensitivity and to allow multi-dimensional experiments. You spin down your cells, you put in your rotor, and then you can run high resolution. You can have high resolution spectra, and I've already quite a lot of information. So this is kind of spectrum we get. And before going further, I will mention that for glycan analysis, the type of experiment we use is called an inadequate, and it's carbon-carbon correlation. But here, instead of the normal single quantum chemical shift you have here, it's a double quantum chemical shift, allowing to have a good dispersion of the chemical shift along the y-axis. And this is very important for glycan assignment, since all the chemical shifts are like considered between 110 and let's say 60 ppm. So inadequate is like kind of compulsory. For this complex system, what is compulsory too is to go to a high field, but also very efficient probe. And this is why we went to the National MagLab facility to access this high resolution and sensitivity. As I mentioned before, starch is a pure polysaccharide, and it's also the simplest polysaccharide in a way found in the cell. It's very easy to detect and to differentiate from other glycan because it's very rigid, and it's the most rigid and crystalline part in the cell, at least in the algae cell. We published like now 4 years ago, a paper mentioning this starch extracted, but also in the cell. And we proposed a methodology that allowed us to identify the type of starch and the quantity of starch directly in the cell. But we also wanted to go further. As I showed you, starch is not the only spin system available in the glycan region. So Using a CCMRD database, which has been developed in Professor Tuo Wang lab at the Louisiana State University, we can have access to a big database on all the chemical shifts of glycans reported in literature using solution and solid state NMR. And if you're patient and if you compare a lot, you can assign quite a lot of different spin systems. Even if they are very similar, not only considering one chemical shift as being discriminant to discriminate between glycans, but considering the whole spin system, you can lead to this kind of assignments. And then using also mass spectrometry, we were able to assign different glycan linkages directly in the cell again, even if we had some overlapping peaks, of course. But after just assignment, which was already nice, we also wanted to have a quantification of different glycans directly in the cell. And this is kind of quantification we had. As you can see here, we had kind of a problem. We had some missing glucose units here. But we realized that running different one-dimensional experiment, that the recycling delay of 3 seconds that was used for the inadequate experiment was too short to be quantitative, fully quantitative. But by running other experiment, we proved that the only species in the algae cells that being rigid enough to be not quantitative using a 3-second recycling delay was the starch. Therefore, we managed to have a correcting factor that were just applied to the starch, leading to this quantification, giving a very nice, very good agreement between mass spectrometry in black and solid state NMR. But solid state NMR also allows us to have other information since it's a native system here. So as I said, it was published just like last year, this methodology about glycan quantification in whole cell systems. So now we propose this system on how we can identify and quantify glycans directly in the cell. We wanted to use the full solid state NMR toolbox to have other information in terms of biophysical properties of, let's say, complex and biological systems. To do so, I will now focus on the Chlamydomonas reinhardti cell wall, which is known to be made of glycoproteins, can be extracted, reconstituted, hydrated, and then studied by solid state NMR. So first, the Chlamydomonas reinhardti cell wall is made of, as you can see here, different layers of proteins that has been described before for several decades now. And those different layers has been described mostly using electron microscopy, but some people proposed and proved that this central triplet shown here can be extracted using a chaotrope agent, very simple extraction. Then you can reconstitute it by simple dialysis. You can freeze dry it before rehydrate it a bit and study it by solid state NMR. Leaving the cell kind of intact because we are not exploding the cell. We just leave the cell surface with few proteins left, but not much. This cell wall is this extracellular matrix of Chlamydomonas reinhardti, making again the algae very interesting to study, is that they survive solid state NMR. It's very hard for eukaryotic cells to survive because with the magic angle spinning, it's very stressing, and the centrifuge and desiccation forces applied to the cells are very strong. But this cell wall is really acting like a shell around the cell, making the cell surviving more than 24 hours without any more media put with the cell at 15 kilohertz magic angle spinning and room temperature. This cell wall is a non-cellulosic cell wall, making a clear difference with plants that are mostly cellulosic based, but instead it's made of glycoproteins. Even if it's not made of cellulose, it's still interesting to compare this cell wall with plants because those proteins has been reported to be extensin related, and extensin are very important, for example, in plants, for growing plants. And those extensin-like proteins are also called hydroxyproline-rich glycoproteins, and those are the main proteins that has been studied in the past decades. They were focusing only on those 3 high molecular weight proteins rich in hydroxyproline, and those previous works mostly reported arabinose and galactose as the glycan units through the O-glycosylation of hydroxyproline. This is what we get in our sample here. So this is a simple SDS page, but we could also make some amino acids analysis on different bands of the gel. Anyway, we proved that we had these HRGPs in our sample, but more interestingly, we also had smaller proteins, and those are rich in asparagine and therefore probably associated to via N-glycosylation to other glycans. And this would explain why in our sample and not previously, high amount of mannose were detected and reported. So as previously mentioned, we will use the inadequate experiment to make some assignment and quantification of glycans directly on the cell wall extract, carbon-13 labeled again. But we'll go further and quantify some part of the dynamic of this assembly using mostly CP buildup and decays. We'll go and quantify the hydration levels using a DAR, water-edited DAR experiment. All that will be done using the high-field and very sensitive probe at the National MagLab facility. And finally, we'll also use MAS-DNP to allow access of the contacts between amino acid and glycans that are not detectable or not easy to detect at room temperature. This will lead at the end of this presentation to a proposed architecture model of this cell wall assembly. So first, as I mentioned and described before, the same methodology. Using CCMRD database, we can assign quite a lot of different peaks. Of course, I won't go into much details, but we can also go to the quantification. But in a glycoproteins, you don't have only glycans, you also have amino acids. So this time you can use the well-known BMRB database and assign quite a lot of amino acids. Again, using similar methodology, we can go further and make some quantification and having a good agreement between mass spectrometry and solid state NMR. But don't forget that with solid state NMR, it's a native sample. So we can have some information on the secondary structure. I won't detail it today, but it's still very interesting information we have here. So this was the previously proposed model with HRGP, so hydroxyproline-rich glycoproteins, linked to glycans rich in arabinose and galactose. But here, we also proved that we have high amount of mannose associated with lower molecular weight proteins, probably linked through N-glycosylation with asparagine. Overall, the spectra represent more than 500 unique peaks assigned and about 30 glycans and more than 80 amino acid spin system, different spin system assigned. After this identification and quantification, we went further to the dynamic. First, we measure the T1 relaxation, proving that overall, and something we have to keep in mind, overall, this assembly is very dynamic. It's nothing compared to a plant cell wall or anything like that. It's not crystalline, it's just very structured, let's say. But we still managed to have some differences in this bioassembly. Here, by comparing the CP buildup and decays, we prove that the glycan has a fast buildup and a slow decay when compared to amino acids that has somehow a slower buildup, but more interestingly, a much faster decay using this CP buildup. This proves that this decay is mostly associated with a long T1 row relaxation for glycans, and this is probing some motion at a milliscale, at a millisecond time scale. So we have less motion in the glycans. So this is the first update we have only with solid state NMR, and it's completely new, showing that we have rigid glycans when compared to more mobile amino acids. Then we wanted to see some dipolar contacts, let's say strong dipolar contacts. So to do so, we use a PDSD experiment, well-known experiment, single quantum this time. And we show that here, yes, indeed, we have some preferential contact between glycans, but by applying longer PDSD, we didn't observe any contacts between amino acids and glycans. Instead, we see all the glycans seeing dead cells, but all the amino acids just disappeared. Being explained by our hypothesis, it can also be followed by the intensity followed for glycans that has a much slower decay when compared to amino acids, where they just don't survive for at 1 second PDSD. So this would be explained by the glycans acting like kind of a magnetization sink, where the magnetization is trapped compared to amino acids that relax during that time. We also wanted to have an idea of the hydration level, because since it's an algae, it's growing in water, so you can guess that water is very important in this type of assembly. To do that, we used what is called a water-edited DAR experiment. Just briefly, it's a T2 filter on the proton, allowing only the magnetization from water to survive, and then we leave the magnetization diffuse and let's say turn on the or make it possible to see the molecules surrounded or having water close to it. And as you can see here immediately, we see that the glycan survived to this type of experiment with the glycosylated amino acid. This is the hydroxyproline here, and leaving the amino acid completely disappeared. This can be, using those spectra, it can be more precisely identified and quantified, again showing amino acid being much less hydrated than glycans. But more interestingly, we also proved that glycosylated amino acids, namely hydroxyproline threonine, serine, and asparagine are much more hydrated, meaning that those linkers have a separated role and dynamic and hydration compared to other amino acids. So this is a new update of our model with very hydrated but rigid glycans when compared to less hydrated but more mobile amino acids, which is a bit counterintuitive, except if you think about water being structurant in this kind of assembly. Finally, we really wanted to see preferential contacts between amino acid and glycans. First of all, as a proof of concept that we can detect in such a complex assembly these contacts between glycans and amino acids, but also in this type of assembly, we know that those linkers are very important and different from other amino acids. As you can see here on the sheared inadequate, so meaning we just tilted the y-axis to make it match a single quantum frequency, the hydroxyproline are first of all very hard to detect at room temperature. But we realized that using inadequate, but here I'm just showing a DAR experiment, just by lowering the temperature without any microwave, we still are able to detect those hydroxyproline, meaning that hydroxyproline had probably previously motion interferences with the inadequate experiment, and lowering the temperature, meaning decreasing the dynamic, made the detection of hydroxyproline possible. Then by turning on the microwave, of course, we increased a lot the sensitivity, even if it was carbon-13 labeled. And this allowed us to detect preferential contacts between amino acid and glycans, finally. We indeed detected the expected arabinose hydroxyproline contacts, but we also managed to see the hydroxyproline mannose contacts that are dipolar contacts and not covalent contacts. So this is the final update of our model. So just as a quick conclusion, we proved that the methodology we applied can be applied to different cells and allow using single solid state NMR combined with mass spectrometry, good agreement in terms of assignment and quantification of several glycans. But like for the last part of the talk, we also managed to detect the hydroxyproline-rich glycoproteins in our cell wall, but we also detected other smaller proteins that are rich and associated with high amount of mannose through asparagine linked to it. We also used solid state NMR to prove the hydration in a native sample, and we also detected glycan-glycan preferential contacts, but more importantly, we detected using MAS-DNP preferential contacts between glycosylated amino acids and glycans. With that, I would like to thank my supervisors, Professor Isabelle Marcotte and Dr. Dora Warshawski, and people in our lab in Montreal. I would like also to thank Professor Tuo Wang at Louisiana State University, and like all his students are here to present during this ENC. So that's all very interesting works. And I also would like to thank people at the MagLab facility, Dr. Ivan Heng, who did the high magnetic field experiment, and Dr. Frédéric Maintil-Viguier, who did the MAS-DNP on remote control during the pandemic. So with that, thank you for your attention. If you have any question, feel free to ask me.
Thanks for a wonderful presentation. So questions? Again, if you can make your way to the microphones.
Are you using same sample preparation for running JJ inadequate at DAR experiments?
Yeah, it's the same sample.
So same hydration levels?
Yeah, same hydration. Yeah, we keep, it's not, when compared to, let's say, a whole cell, you can say it's not that hydrated because it's a 2-to-1 ratio. Like we have twice the volume of water for 1 volume of cell wall. So it's, let's say, a dense sample. But if you think about in the cell, locally, it's not that hydrated. But yes, we kept the same hydration levels.
So for these samples, do you have like a MAS limit beyond which you don't want to spin? Because usually you want to keep the spinning speed lower, right?
Mm-hmm.
So what is your cutoff limit kind of thing?
So for us, we even try 26 kilohertz. That was our technical limitation in our lab. But still, instead of 24-hour survival that we had at 15 kilohertz, at 26 kilohertz, so in a 1.9 millimeter probe, we had a survival of about 10 hours, which is enough to have, let's say, one spectrum if it's labeled.
So how do you define survival? Is it from the perspective of the spectrum or from?
No, we take out the cells and then we make some viability tests based on fluorescent assay and flow cytometry. Because we have the chlorophyll that is fluorescent, but you can also add other molecules that can be metabolized and prove that you have living cells in your rotor.
Thank you.
Any further questions? Well, then join me in thanking Alexandre again. Our next speaker is Jacob Holmes, who, as the title of his presentation, is Proton Positions, Positional Uncertainties, and Inhibition of the Trypsinthase Aminoacrylate Intermediate.
I'm sure everyone asks this, but can you hear me? Yes, okay, thank you. So hello, everyone, and thank you for having me. Today, I want to talk to you about some of the work that has been going on in our lab. I'm going to be focusing on the tryptophan synthase active site, where we were able to place hydrogens, determine positional uncertainties, and learn about the chemistry. There are many methods which give us structural information about a protein or enzyme system, and these methods can offer indirect information about the chemistry of our system. I'm going to be talking about work that has been going on in our lab where we can improve our structural detail, but also gain insight into the chemical environment and the reaction mechanism. Indeed, a motivating factor for us is knowing the chemical environment and reaction mechanism. To do this, we need to know the position of our hydrogen atoms. But beyond their position, we need to know the uncertainty of it. And we do this by extending an approach from Lyndon Emsley's group, which gives us anisotropic displacement parameters for atoms in our active site. For this work, we're using what we call NMR-assisted crystallography, which is the integrated approach of X-ray crystallography, nuclear magnetic resonance, and first principles calculations. All three are powerful tools in their own right, but by combining them, we gain resolution greater than any one technique. The system we are looking at today is the beta site reaction of tryptophan synthase, the cofactor pyridoxal 5-phosphate, shown in black, and in the resting state is bound to lysine 87, shown in blue, through a shift-based link. L-serine enters the active site. L-serine enters the active site, and it displaces lysine 87 to form a new shift-based link between the PLP and L-serine. The beta hydroxyl leaves as a water, and we form the aminoacrylate intermediate. Indole, also shown in red, comes down from the alpha subunit and forms a bond at the C beta of serine. This product goes on to leave as L-tryptophan, and we return to the internal aldemine or the resting state. I want to highlight a few of the intermediates which we can study through NMR-assisted crystallography. Some of these have been published, but some of these are ongoing work. I also want to point out that these are catalytically active intermediates, and the lifetime for some of them can be short. For today's talk, I'll be focusing on the aminoacrylate boxed in red. Our starting point is to solve an X-ray crystal structure because it gives us a preliminary view of our enzyme. This list is some of the crystal structures we have solved in our group and with our collaborators. These are great starting points because we can begin to infer hydrogen bonding networks and measure distances around the active site. But we want to make more definite statements about the chemical environment. For that, we need to be able to place hydrogens and do it with high resolution. One method which exceeds at delivering details about the chemical environment is nuclear magnetic resonance. How we get this information in the active site is to introduce isotopic labels like L-serine. L-serine is the natural substrate of the aminoacrylate, and it allows us to collect these three carbon shifts as well as the shift-based nitrogen. Our synthetic collaborators are able to synthesize PLP with the following isotopic enrichments. The enriched PLP is incorporated into our enzyme, and we can get these shifts along the ring as well as the purity nitrogen. For beta lysine 87, all lysines are uniformly nitrogen labeled, but here we can take advantage of two things. The lysine is close enough to the phosphate group, making it a great candidate for REDOR. But we also see a unique lysine shift that is separate from other lysines, and it matches the time scale of our intermediate. Here are a few more of the spectra we have collected, which include O17 shifts for the carboxylate on serine. One thing I want to point out now is the temperature dependence of the shift-based nitrogen from near room temperature all the way down to 95 Kelvin. So with an X-ray structure in hand and some preliminary chemical shifts, we can move to first principles calculations. We're using a cluster-based approach to computationally model our protein active site. Using heavy atoms from the crystal structure, we take all residues within 7 angstroms of the cofactor. We then place hydrogens on the residue and the cofactor. The protonation states of the cofactor and substrate are unbiasedly explored. So the 7 angstrom cuts are geometry optimized with the outer layer fixed, holding the heavy atoms in their crystallographic locations. Once the models are geometry optimized, we can perform NMR calculations and get isotropic shielding values, which can be converted to chemical shifts using the linear rescalings listed. For a correct structure, we expect from model compound studies to have the error ranges listed: 1.5 ppm for carbon, 4.3 ppm for nitrogen, and 7.5 ppm for oxygen. For the aminoacrylate, we modeled 35 different combinations of protonation states. These are again done in our 7 angstrom cuts and allowed to geometry optimize, and then NMR calculations are performed. What we need now is a way to evaluate when a model is able to reproduce the chemical shifts well enough that it gives us confidence the model reflects the proper chemistry and reactivity. The first step to doing that is using the reduced chi-square statistic. The models are ranked according to their reduced chi-square value, which is the weighted deviation of the models from the experimental shifts, and whether they meet the 95% confidence limit. But before I jump into the results, let me take a step back and talk about what components make up the reduced chi-square. Here, N is the number of experimental NMR shifts we have, which include isotropic chemical shifts, as well as any chemical shielding tensor components. F is the number of adjustable parameters, and for our initial screen is just 0. S is the RMSD from model compound studies and is dependent on the nucleus being modeled. For 11 shifts, the confidence limit is 1.79. We choose our cutoff to be 95% or choosing an alpha value of 0.05. This says that if we have the correct structure and the expected precision to calculate the chemical shifts that give us the RMSD in model compound studies, 95 out of 100 times we would find a reduced chi-square below our confidence limit. If our value is above this confidence limit, we can reject the model with 95% confidence. Getting back to our rankings, we see that no single model falls below the confidence limit, and we must begin to look for another explanation which describes our data. For this, we keep in mind that the shift base is temperature dependent. And so motivated by this temperature dependence, we consider fast exchange equilibrium models between models that differ at most by the position of one proton. We do this in an unbiased way, so we form all possible pairs and optimize the populations that minimize the chi-square. The predicted chemical shifts now become the population weighted averages of these two models. The population weighted averages are compared to the experimental shifts, and we see that the reduced chi-square are smaller for equilibrium models when compared to single site models. With so many combinations, we expected more than one might fall below the confidence limit, but in fact, only one unique exchange satisfies the 95% confidence limit. Shown here separately for the isotropic chemical shift, as well as the chemical shielding tensor of the shift-based nitrogen. The winning model is a proton exchange between the shift-based nitrogen and phenolic oxygen. And this is consistent with the shift-based temperature dependence observed in the nitrogen spectrum. What I want to do next is say how much better the winning model is over competing models. And for that, we turn to Bayesian analysis. Using Bayesian, allows us to improve upon our current assumptions and provides a measurement for how much more likely one structure is over another. This was a nice way of analyzing the probability which Lyndon Emsley's group introduced into NMR crystallography. The first step is to calculate the relative probability compared to the winning model. We do this using the F distribution, which describes the ratio of two reduced chi-squareds if both are equally good descriptors of the expected of the data with the expected residuals. The probability of M is the prior probability, which we can use here when a model is consistent with observations not included by the reduced chi-squared, such as the temperature dependence of the shift base. From Bayesian, we can see that our best fit model from the reduced chi-square is most probable with an 88.5% probability. From the analysis, we can move from saying we are looking at models to saying we have a 3D chemical structure. We were looking at the amino acrylate of tryptophan synthase. For this, we find a proton exchange between the phenolic oxygen and the shift-base nitrogen, whose populations were determined to be 89% protonated phenolic oxygen. The pyridine nitrogen was found to be deprotonated, which in earlier proposed mechanisms was thought to be protonated. The acid-base catalyst, lysine 87, is neutral, and I will talk more about the catalytic importance in a moment. But before I talk about the catalytic significance, let's talk about how good the structure is. To do this, we are using another method from Lyndon Emsley's group, which was applied to small molecules and now extending to our structures of tryptophan synthase. The method tells us information about the positional variance of atoms in the active site, which include hydrogens that we have placed through NMR-assisted crystallography. The aim is to generate a large number of chemically reasonable structures, meaning they don't have unrealistic bond lengths or bond angles. To do this, we use low temperature molecular dynamics to generate the structures. In the scatter plot, we have about 10,000 structures which have their positional deviation in the principal axis system of atom displacement, plotted against the square root of the reduced chi-squared of the chemical shifts for the perturbed structures. What we get is a series of expanding Gaussians of the atom displacements at a chi-squared value. You'll note the three lines which represent 1 sigma, 2 sigma, and 3 sigma, which are the variation from a Gaussian distribution. And so we claim with 95% confidence, structures beyond a reduced chi-square of 1.77 is inconsistent with the experimental NMR shifts. And so that is where we'll draw the uncertainties. And so what does all of this look like on a structure? Well, we find that the RMSD of hydrogens, which we have placed through NMR-assisted crystallography, is 0.17 angstroms. And for heavy atoms, we find that it is 0.11 angstroms, which is 6.5 times smaller than the equivalent structural uncertainties in X-ray diffraction. I'll note here that the ellipsoids shown are with 95% probability. So with our structure in hand, what can we say about the chemistry of our system? Well, here we see the amino acrylate in the center picture. It is remarkably close to what we are calling the preceding transition state. The charged lysine donates a hydrogen to the beta hydroxyl, making it a more efficient leaving group. The beta hydroxyl leaves, and the newly formed water is caught in a hydrogen bonding network. We're going to call this a weak binding pocket, which is consistent with a minor peak in NMR. We can also react it with the inhibitor shown on the right panel. And for time's sake, I will just comment that it was very interesting to learn why the inhibitor does not proceed with the bond forming step, despite being a better nucleophile than the natural substrate. The key find from NMR-assisted crystallography is that the lysine is positively charged here, and I'd be happy to talk more about this intermediate, would probably be another talk on its own. To wrap up here, I want to summarize what we were able to do when we combined X-ray diffraction, NMR crystallography, NMR, and first principles calculations. A huge takeaway was that we could place hydrogens in the active site with an RMSD of 0.17 angstroms. With the hydrogen atom positions, we could start looking into the chemistry and reactivity of the amino acrylate. We determined a proton exchange between the phenolic oxygen and the shift-base nitrogens, whose populations we could determine for each tautomer. The last point is that our resolution was great enough to see a weak binding pocket, which was tenths of angstroms away from the preceding transition state. Most importantly, this work emphasizes chemistry, which has always been NMR's greatest strength and where we think it will integrate most strongly with other tools of structural biology, including X-ray diffraction, cryo-EM, and even neutron diffraction. I want to show a quick acknowledgement slide to all the people who aided in this effort, both current and former lab mates, as well as our outstanding collaborators. And I'd be happy to take any questions.
Thanks, Jacob. Questions for Jacob?
Thank you for the nice talk. I was just wondering, have you considered using QMMM approaches as an alternative to the cluster approach?
So the whole idea is to get it to be purely QM. So we haven't looked at that. Yeah.
Thank you.
Any further questions for Jacob? Okay, then let's thank Jacob again. Okay, our next speaker is Ruth Stark, and the title of her presentation is MAS NMR View of Melanin Pigment Deposition in Fungal Cell Walls, Laying the Foundation and Greasing the Wheels.
First of all, thanking the organizers for giving me this opportunity. And I'm going to start by acknowledging the contributors to this work so that I don't forget at the end. And they include recently graduated PhD student Christine Christian, postdoc John Kelly, our NMR manager Xin Wang, who I believe is in the audience, and our collaborators at Johns Hopkins, Arturo Costa-Deval, and Emma Camacho. And of course, those agencies that pay the bills. But the star of the show is this melanized fungal cell. And what I want to emphasize here is that melanin insinuates itself in the cell wall, and then there is an outer capsule in this particular type of fungus. And what I want to focus on today is not the melanin per se, although I am going to talk about it. I want to focus on the scaffold, that's the foundation, and the rather unexpected lipids that play a role in this process. So why are we interested in this problem? Well, melanins are heterogeneous aromatic indole-based polymers of still unknown structure, and they mean a lot of different things to different people. To the fungal cell, they are protective agents, but they are also, and is greatest interest for us, they are virulence factors for various kinds of infections, which can be quite dangerous. But if I think about melanin as this protective agent, it has to get into or onto, it has to deposit onto the cell, and it has to adhere. And so basically it takes two to tango, or maybe three to tango, and that's what that's the premise of the talk today. So I want to say a little bit about how the fungus makes this material and how we make it. And it's important because the fungal cells, particularly Cryptococcus, must scavenge or be fed these catecholamine precursors such as L-DOPA in order to melanize. That is, glucose, not interested, glycine, not interested. They need these particular precursors. And if they have them, we will get what I've shown here in cartoon form, a melanized cell. And what we typically, but not always do, is to do a series of enzymatic and chemical manipulations in order to isolate the melanized cell walls. And those are called melanin ghosts. And I will show that as I go along. And then I will back off and show some other ways to look at it. Now, these materials are insoluble, not well ordered, and rather heterogeneous chemically and also motionally. And that does make them challenging to study. But the part of it that I want to focus on in this talk is not the melanin per se, but the scaffold that it has to lie down on in order to serve its protective function. And an example of the importance of that is shown here. This is work done by several biologists from Jenny Lodge's group quite some time ago. And the observation is that the precursor glicnac and acetylglucosamine, if you prevent that from forming the corresponding polymers, which would be chitin, represented by this monomer, and its deacetylated form chitosan. This is not a copolymer, but I've just shown the monomers that way. If you prevent that from happening by genetic manipulations, what happens is that the melanin is just fine to form, but it does not adhere. It leaks out into the cell medium. And that is generally called leaky melanin. And it emphasizes the importance of the scaffold as well as the pigment. And so spectroscopically, even at natural abundance carbon, what you can see is that in this kind of wild type cell, I can identify all of the major carbon types. These guys are polysaccharides, these are lipids, we'll talk about them more. And this lump underneath here is the melanin. And clearly there is less of it in a leaky melanin sample because it's no longer in the solid state, it's off into the cell medium. So we wanted to understand what's happening there and what the role is. And so if we wanted to focus on the scaffold, what we did in that case was to do isotopic labeling with glucose. And that enriches everything except the melanins because they can't eat that material. And so if we do that, just even in 1D, we can compare sets of wild type and leaky melanin ghosts. And these are Cryptococcus neoformans. These are a different species, Cryptococcus gotti, and we've got those pairs. And what I've marked off are the different classes of compounds. And because we did this with direct polarization and a long recycle delay, we can figure out how much of each class we have. And what we see here is that the lipids, rather surprisingly, are dominant. And the reason that's surprising is that to make these ghosts, we need to do 3 successive lipid Fulch extractions and an hour of boiling in concentrated HCl. And it's a very harsh treatment. And yet these lipids survive. And the polysaccharides, which are what you'd expect in the cell wall, are diminished, but some of them do survive. And then there are other compounds of various other classes. So that's kind of the lay of the land. And so what I want to focus on first are the polysaccharides, the so-called scaffold, and then I will come around and say something about the lipids. So the polysaccharides can be a lot of different things, but our interest was in the chitin and chitosan because in the leaky melanins, those are the guys that are being knocked down. And so what we did to focus on them was to do Z-filtered TDOR experiments. So this was labeled with both carbon and nitrogen. And these are actually 2 overlaid spectra, each one optimized for the number of bonds, really the distance that we are sampling. And we see right away that the nitrogen-containing polysaccharides, the chitin and the chitosan, just stand out, can be distinguished, and we can identify all of these peaks. And this particular one tells us that the chitosan is highly deacetylated, and that's the lay of the land. We also get from these spectra a map of what the carbon shifts are from those 2 polysaccharides. And that's important for what I'm going to show you in just a minute. And that is, we can do the corresponding 2D DAR spectra of these materials and try to ask, how do the 2 strains, the Cryptococcus neoformans and the Gotti, how do they differ from one another? And how does any wild type differ from the leaky melanin example? And what we see here, first of all, is that the general patterns are similar, but because we know those carbon shifts from the TDOR, the anomeric carbons can be distinguished. I can identify separately the chitin and the chitosan. And although the patterns look similar, if you look closely, as we'll do in a minute, you can see that some of the peak intensities differ. So this is a rather short DAR intentionally. And what are these differences? So what we did was to look at the slices here because it makes it easier to see actually. And here's what we have. The first comparison that I'll make is between the 2 wild types, okay, where I have neoformans over here, Gotti over here. And this is the diagonal. These are the off diagonals. And you can see, particularly in this bottom one with chitin, that it's the same peaks, but both their line widths and their relative intensities with respect to the diagonal are changed. So that's basically telling you that the dipolar couplings or the averaging of the dipolar couplings are different. The degree of rigidity and order differs when you have one strain versus the other. Now, if you take a look similarly, analogously anyway, at wild type versus leaky, what you see here is that these guys drop down in the leaky melanin. And that means that you can imagine that it became too disordered and it's leaky. In the same way, for the Cryptococcus Gotti, same trend, maybe not as dramatic, but too disordered. If you move on to the chitin, it's a rather different picture, not as clean, in fact. In this case, with the neoformans, the leaky one again looks too disordered or to adhere, but this goes in the opposite direction. So the point is that the scaffold matters, and it's the chitin and chitosan that matter tremendously. And it turns out that they are the principal polysaccharides that survive the preparation to make these ghosts. So these are the most important. Okay, so that's a vignette regarding the scaffold. The model that my student likes is this mud hut. The idea is that if you want to slap a pigment against that wall, it matters what the organization of that wall is. And that's what the fungus is doing. Okay, so in the last couple of minutes, I'm going to return to this diagram, this quantitation that I showed you earlier, and talk a little bit about the lipids, because it was, in fact, surprising to us that the major constituent that survives the preparation of the ghost is lipids. You would think that they would be gone, and that's not what happened. And so there are a lot of ways to look at them, and I'm going to show you a few. The one that we've already seen are the ghosts, okay? These are the melanized cell wall. So it's made from this preparation to make the ghosts. Now, if you do that, what happens, in fact, is that the polysaccharides are diminished and they have a somewhat similar, at least comparable intensity to the lipids. They're not going to wipe them out in dynamic range terms. But that's a lot of perturbation that you really don't want. But you can look at the whole cells, and there are a couple different ways you can do it, some of which are like one of the prior talks in this session. And the idea is that if you look at a whole cell system, then you can compare melanized and not, which you can't do with the ghost because you can only look at the melanized cell wall. And if there's no melanin, you have nothing left. And furthermore, you can look at these guys wet or at least damp, and that gives you a near native kind of preparation. Now, if you do that, the resolution is magnificent, but of course the system is a bit more dilute. So that's a bit of a trade-off. And these kinds of approaches have been used a fair bit during the last decade, and it's also something that we tried almost 30 years ago in a different context. So many ways to go about it. And what I want to focus on is what is going on with the lipids now that we've dealt with the polysaccharide scaffolds. And what I want to take you through is, first of all, if I take a CP mass spectrum, these are labeled. This is a strain without a capsule, as it turns out, but these are dry whole cells. And it's not really so surprising, I think, that the lipids do not cross-polarize very well. But that means we can turn this around and say, well, they're motionally quite mobile, and we can do a short recycle delay on a direct polarization, and lo and behold, the lipids jump up, and the resolution of all of those is magnificent. And what that means is that we have entree to a direct polarization inadequate experiment, where now we're getting carbon-carbon through bond couplings. And in fact, we can identify by reference to the published literature what these guys are. And one example of that is that all of these that I've marked off tell us that we have these two particular sterol esters, neutral lipids esterified. And furthermore, we can complement that experiment by doing, again, a liquid-like preparation combined with a heterocorrelation proton-carbon through space in this case. And between the two, we can establish that we have not only the sterol esters, but we have these polyisoprenoids and dominantly, quantitatively, triglycerides. That's what we have in there. And the first kind of question to ask is, does this require melanization to be present? And the answer is no. They're there anyway, and they may facilitate the melanization, but they are there. Secondly, are the lipids retained in melanized cells? Well, they are. We wouldn't see them otherwise. And that they are retained preferentially to the polysaccharides. And that really is surprising. And moreover, the sterol esters and the polypropanoids are retained preferentially. Now, what we don't know yet, and I present it as an open question, is are these guys, what are they doing there? Are they readying the cell for the production of melanin, which will produce reactive oxygen species that you might want to scavenge? What's the deal? And I leave that as an open question. All right, so these are my conclusions. First of all, what matters in melanization is not just the production of the pigment, but also what it is depositing on, namely the scaffold. We can probe that with a variety of experiments ranging from direct polarization, MAS, to TDOR, to DAR. They're all informative. to tell us how the leaky melanin varies with respect to the wild type. And we can identify and quantitate all of these surprising lipids that are also present and somehow playing a role. And finally, solid state NMR can bring us closer to native systems, although we're not quite there yet. That's to be continued. So that's where I'll stop, and I'm happy to take any questions.
Thanks, Ruth, for a wonderful presentation. So questions for Ruth? Did I miss, can you tell where the melanin is binding on the cell wall, like precisely?
Well, you didn't miss it because I did not talk about it. And we can tell if we label the melanin as well,
Yeah.
and we have done that, although I didn't talk about today, we can implicate the chitin or chitosan through the carbon, which at the time we could not distinguish, okay? But we know that those chitinous, well, I'll call them chitinous materials, are the ones that are closest to certain parts of the melanin. And that's about as far as we are right now, in part because it is not possible to choose where to put all the labels on the melanin precursors, okay? So we can, but not fully, would be my answer.
Further questions for Ruth? Well, then join me in thanking her again. Okay, our final talk this afternoon is by Ben Wiley from Texas Tech. And the title of his presentation is The Functional Interplay Between Lipids, Channels, Receptors, and Biological Assemblies as viewed by solid state NMR. Ben?
Thank you. And actually, I'm having mercy on you. I'm restricting myself now to just cholesterol, or mostly just cholesterol. I'm standing between you and bittersweets. So I'm going to tell you about how cholesterol is able to functionally regulate two classes of proteins. And so the first one we're going to talk about is going to be classical Kerr channels. Now, my stand-in for classical Kerr channels is going to be this prokaryotic channel called Kerbach 1.1. I'll tell you about in a second. But these channels are all going to be anionic lipid activated, or anionic lipids are some way going to be involved in their activation. And cholesterol can either have a competitive or a cooperative effect with these anionic lipids, depending upon the class of the Kerr channel. So that's actually kind of interesting that these channels can have such diverse responses to cholesterol. But these classical Kerr channels tend to be inactivated by it, which is interesting because they exist in cholesterol-rich membranes. So then why would you have something that's inactivated by a native lipid? I'll make sure I stay on time. Then next we're going to talk about, I'm going to tell you a little bit about our work on CCR3. Now these proteins, this would be a chemokine receptor, so it's involved in our immune response. It also has a relationship with anionic lipids, but we're going to focus on how cholesterol sort of primes it for activation by its native chemokines. All right. So first, I want to tell you why it is we care about what cholesterol is doing with the Kerr channel. And for that, I'm going to tell you briefly about the activated state. All right, so Kerr channels are like most potassium channels. They have an extracellular gate that is able to dehydrate and then rehydrate potassium to allow it to pass into the cell. And all these channels are going to be opened based upon some sort of activatory stimulus. In this case, it's going to be anionic lipids that are going to bind here at the cytoplasmic interface. That's going to cause the channel to open. Okay, this is fundamentally important for the resting membrane potential, which is to say the launching point for the action potentials in our bodies. If we don't have that proper resting state, then we're not going to actually then have this cascade of effects that we call an action potential. Okay, so Kerbach has a similar fold to most Kerr channels, except it has a cationic binding pocket that is optimized for things like PG, right? Relatively small anionic lipid head groups. And you can see that this electrostatic map of Kerr channels, which is pretty conserved across types and species, has this really large cationic pocket that is right below the inner leaflet of the membrane. All right, so this is where the anionic lipids are going to diffuse in and then allow the channel to activate. We do have near complete chemical shifts of this protein, even though it's over 330 residues. And so to show you I'm not going to tell you all about this. This was earlier work, but we do have chemical shift assignments that are well enough to tell us that we can see the open versus closed state of the channel, which is to say, when we have the protein in just switteronic bilayers, we can see it closed, and then we can actually see it activate here in red. And we're going to actually talk about how cholesterol promotes the green state that you observe here. Now, we are working actively on the activated state and anaerobic lipids, and Reza Amani, my student, will be glad to tell you about that at poster two. And I should say we are having a lot of progress getting the channel to open. You can see here our preliminary open state of the protein. We're actively working with the PAST algorithm with Charles Schwieters at NIH to basically get all the right intermolecular contacts to get the native open state structure. Okay, so cholesterol. All right, so we first started looking at Kerbach as the way that it was actually able to coordinate lipid behaviors in its native membranes, which actually contain this 5-membered ring called hopanoids, okay? And so these hopanoids appeared to group themselves near the protein. And so we started wondering about this because we started to see that there was actually these cholesterol binding clefts that were located in Kerbach 1.1 that were very similar to the classical KER channels, KER2, which are found in our heart and our central nervous system. And we started to realize that this is actually a fairly common model system to look at cholesterol regulation KER channels. And so we started looking at what it would look like in our hands. And so to do this, we adapted a bunch of assays and FRET measurements. And so first what we like to do is this fluorescence quenching assay. And in this particular experiment, we reconstitute our protein in the liposome. So everything we're solid state NMR, everything we do is going to be in a liposome environment. And so we reconstitute it into liposomes. We introduce a fluorophore that is pH dependent. And we then allow that to incubate. And then we introduce an ionophore that brings in H+ cation into the center of the liposome, pushing out potassium. So as we can watch, as our fluorophore quenches, we can also basically measure potassium flux using that. As cholesterol increases, we see a small bump in activity at low cholesterol levels, but then as the cholesterol mole percentage increases, we start to see the channel close. This is then reflected in FRET measurements where we can see that when we have the gated state, as we start to add cholesterol, is then actually shut off. So of course, we're an NMR group, we want to do NMR. So the Howard Riesman at the University of Geneva gave us a gift of his yeast that is able to make cholesterol. And so we adapted a skip label cholesterol scheme that was published by Della Ripa and Chad Rienstra some years ago. And using this, we were then able to, using either one or two C13 labeled acetate, we're able to introduce this skip label cholesterol. So we're able to then have cholesterol that has really nice resolved carbon lines, skip labeling, so we can then basically do DARB distances much more quantitatively than with uniform labels. We then also grew Kerbach using glycerol, is the way that was first introduced by Lemaster, and of course Mei Hong and Hartman Ashkenat introduced this to solids NMR a couple decades ago now. And this basically gives us the same kind of skip labeling that we're getting in the cholesterol, which is going to facilitate distance measurements. So the first thing that we noticed when we put cholesterol together with Kerbach in these liposome samples is that the cholesterol actually did not give us the same spectrum as it did when it was just in bilayers with POPC. So here in black, we have a 250 millisecond DAR spectrum of our cholesterol samples in POPC bilayers. And here in pink, we're actually able to see, or I guess this is more purple, but we're able to see that those peaks actually all split. They broaden and they actually come into doublets. Okay, and they form these asymmetric doublets. And after further analysis, we discovered that those doublets actually, as we continue to mix out to 1 second of DAR, we started to see that these doublets were actually in contact with each other. And lo and behold, as we started to aggregate all these distances, we found out that we had a cholesterol dimer. Okay, so the cholesterol dimer was present in our sample. And then on further analysis of that dimer, we found that it was actually in contact with protein. And because we had two environmentally distinct cholesterol species, we were then able to determine which face of that dimer was present in our sample. I should actually mention here real quick, this dimer, because there's a lot of discussion and controversy about how cholesterol dimers form, we're seeing what would be considered a smooth to smooth dimer, where the smooth faces of the cholesterol coming together, leaving this rough edge where you have the methyl groups protruding out. So looking at the pattern of distances between our protein and this cholesterol dimer, we were then able to recognize that it was bound to the protein. We then went to explore NIH. As I'm sure many of you are aware, right, Explore is actually going to be a fork of Charm, so you can actually update Explore on the back end with all sorts of functionality from the current Charm distributions. And so what we did is we took and added and generated all the cholesterol topology parameters and force fields we needed, introduced that into Explore NIH, and we're able to generate that cholesterol dimer structure. Then we took the crystal structure of Kerbach, we modeled in the loops that were missing using the Swiss model, minimized that in NAMD, and then we performed simulated annealing calculations between the cholesterol dimer and the protein. Okay, and that was really useful to use these these implicit solvent functions I'll tell you about in a second. And from that, then we were able to get a structure of the dimer docked to Kerbach. And so if we go here, you see what we initially did is that we had a family of structures where we took the 10 lowest energy structures and the 10 lowest energy structures based on the NOE distances. And they gave us a total of 14 structures, but they actually had 2 different orientations. So at that point, we included EFX and IMMX, which are these implicit solvent potentials. EFX is taking care of water. IMMX is going to be the membrane. These were originally, so these I think came from Charm 22, but they were put into Explore NIH by the group of Francesca Marassi. And so once we applied those semi-empirical force fields, then everything converged to one solution. And of course, that has to do with the fact that the OH on cholesterol is going to want to find the water, right? So we got this, we saw the structure of this dimer in this relatively upward state, about 16 degrees tipped from the median. And then doing this, we were then able to analyze, after doing all the simulated annealing, we were able to analyze this particular site. And looking at the pattern of methyl groups and aromatics, especially the aromatics near the base and the aromatics near the top, we were able to find that this site, if you're going to consider that, the 3 classes of cholesterol binding motifs that are often talked about, CRAC, CARC, and CCM, this actually, strangely enough, had this most in common with CCM, which of course is not that surprising considering it's the cholesterol binding motif where we first identified cholesterol dimers. And this was back in the beta-2 adrenergic receptor about in 2008. So of course, this is interesting for us. And the reason that this is actually relevant is because this particular pocket on the protein happens to be nearly identical to what you find in the classical Kerr channel. So it appears that this is what is the native inhibitory mechanism for cholesterol in these channels, is that it turns out the arginines that are required to bind ionic lipids are located almost directly beneath this particular binding pocket. So a cholesterol dimer is able to form, slide in, it competes competitively, or I should say that's redundant, it competitively inhibits the binding of anionic lipid and thus is able to bring these channels to a close, right? This is probably so efficacious because it is a dimer. So you have 2 lipids forcing out 1. And so, these were similar to things that had been observed in GPCRs. And so that's my not so subtle segue into our work on GPCRs. And so we're looking here at CCR3, which is a class A GPCR. It's a chemokine receptor. And we picked it because it happens to be one of the most promiscuous chemokine receptors out there. There's not been a single drug trial that's actually been successful with this receptor. Everything that hits it hits something else. And it also happens to be a promiscuous receptor for all kinds of other things. But it's heavily involved in cancer metastasis, inflammation, HIV, and most recently, it's one of the chemokine receptors that's that's been identified in the chemokine storm with COVID-19. So, right, these severe, immunoresponses so many of us have to COVID viruses, where we're having to take heavy steroids. This is one of those receptors that's actually being over-triggered by that storm, causing all these inflammation responses. So the first question we want to ask, right, we have C13 labeled cholesterol, we want to use it, but we want to basically confirm that this receptor has a similar response to cholesterol as other receptors. And so we started to develop 2 assays. One is fluorescence polarization. Here we have a fluorescent tag that is attached to our chemokine. This is the CCL11 chemokine. And then we're going to titrate in liposomes. So everything we're doing here is in liposomes. We had SMALPs. The SMALPs did not keep all of our cholesterol in the disk, so we stayed in liposomes. So we titrate in the liposomes, and we find that as the cholesterol concentration increases, so does fluorescence polarization, leading then to, or I should say, indicating that the KD is lowering, which means the affinity is going up. right? So ligand affinity is increased by cholesterol concentration. And likewise, we adapted an assay that we first saw used by Scott Prosser, where we expressed the G alpha inhibitory subunit, and then we used that to measure GTP turnover. Okay, so we found then once again, and we once again did this all in liposomes, which required, you know, careful reconstitution with 3 different proteins. But then we were able to find that this GTP hydrolysis, where we convert the GTP to ATP and then measure the luminescent signal, that hydrolysis actually also increased as cholesterol increased. And I should point out this is mole percentages, not mass percentages. So as cholesterol increases, so does signal transduction. All right. So that, of course, is like one of the big things we want to study, right? We want to do all this detailed stuff we've done on Kerbach, but we want to do it in less time. Okay, so you know, all this, we have structures of Kerbach in the closed state and the open state, but it took us 5 years, right? I'm kind of getting impatient now. And so then we started thinking about how could we improve our workflow. And of course, there's all this stuff about, you know, thinking about how, like it's went on for, you know, a couple of decades. How can we improve our workflow by basically modeling things, assigning our data based upon those models? But then we started thinking, okay, we're doing all this molecular dynamics. We've been doing molecular dynamics pretty heavily since COVID. It was something we could actually do at home, thanks to NMR Box. And so we started messing around with Compass. Okay, so Compass is a program that was written by Joseph Portney when he was still with Chad Rienstra. And what it does is it's able to score predicted shifts from a structural model against experimental data. So you see here one of our structural models, the predicted shifts in black, our experimental spectrum here in purple. We're drawing this up to 9 times the noise threshold. And so what we did is first, you know, we templated a whole bunch of models, and we were able to then score all those models using Compass. And then from that, we were able to figure out which is our best template. All right, so we were able to template multiple models of GPCRs based on this, and this, I should say CCR3 does not have a structure, right? So we had to first get something to work with. And we first went through, scored everything based on Compass, and then we also went back and looked for what models were actually more complete. Where do we actually have all the loops? Where do we actually have well-defined structures? And so we decided upon this model that was templated off the related CCR5 chemokine receptor. And so then we started wondering, okay, could we actually use this method to figure out what the protein is actually doing in the bilayer? And so what we did is at 37C, which is the same spectrum, we took our spectrum. We did perform 4 atomistic MD replicates. 4 of them were going to be in just a POPC bilayer, and the rest are going to be including, you know, would be 30% by mass cholesterol. And so we took our output model, we predicted the shifts in Compass. And so from this, we got this spread. So in this, we've got the Compass score, which you can see the POPC simulations, not surprisingly, have a much wider spread because they're not as conformationally selected as the cholesterol, which relative to Compass is going to be a much narrower overall range. But then we have this RMSD versus the starting structure, and you can see for a lot of our cholesterol simulations, it consistently starts to creep up. Now, the average Compass score difference for cholesterol is better than it is for POPC, but we're only talking about 0.3, 0.4 ppm here, which of course at my point, when I first looked at this, I thought, okay, so we're able to say we can actually model GPCRs now and score them, but this is, you know, other than that, this is kind of a boring exercise. But then my student showed me what these models looked like coming out of the MD. And the first thing we started to notice is that for most of the top scoring models coming out of the cholesterol simulations, we started to see better structure for activation in some of these external loops compared to the POPC structures. Likewise, we actually started to see intracellular loops that appeared to be slightly better poised in order for signal transduction compared to replicates in the POPC bilayers. So we then went and looked at the top scoring replicates for Compass using each of these trajectories. And what we found was that indeed this actually bore out that the best scoring trajectories from the cholesterol-rich bilayer were actually appeared to be better based on what we knew about the steps of this chemokine signal transduction than the POPC. So the first thing we noticed is that this extracellular loop 2 is going to be is consistently better structured in all of the cholesterol, in all the scored cholesterol trajectories, and also seems to have the right fold and orientation in order to basically be ready to recognize that ligand, right? This loop is involved in recruiting its native ligand to bring it into the orthosteric pocket. Likewise, we see ECL3 also is in the proper orientation in all of these top models, which is also important for chemokine recruitment, chemokine binding. But what then, so that this in itself was a little bit interesting, but what was really actually dramatic here was that if we looked at the orthosteric binding pocket, which in the case of these receptors, you have to have 2 tyrosines and a glutamic acid come close to each other in order for to have the second step of signal transduction. We found that the average contact scores for these 3 residues in the cholesterol-rich bilayers was significantly greater than in the POPC-only bilayers. And then if we actually started to look at those models in the PC-only bilayers, the average contact score was 1 with a top score of about 9. Whereas when we added cholesterol, the average score was 6 and the top contact score was about 22. So that we actually have a significant improvement of basically the priming of this receptor for chemokine activation when cholesterol is present in the membrane, right? So of course, this is just the starting point for our sort of iterative data analysis, which is going to be ongoing as we acquire all the spectra to assign all the chemical shifts. And one of the other things that we're also going to be focusing on is not only using C13 labeled cholesterol for distances and the like, but also thinking about using it for dynamics and orientation studies. And so one of the things we've worked on recently is introducing fractional deuteration using deuterated acetate. So basically the same pathway, this molybdenate pathway that allows you to bring in acetate to do the fractional labeling using C13 of cholesterol actually will also allow you to do fractional deuteration. So you can then introduce a fair number of deuterons, and using things like respiration CP then, you're able to actually site-specifically look at the sideband manifolds of each of these sites in deuterium. So, bringing this all together, we're really excited about all of our future work looking at cholesterol oligomerization and protein functionalization. So I'm exactly on time. I'm excited. All right. So thank you so much for your attention. I would like to really, a lot of this work was actually done by the Kerbach work was heavily done by Colin Borsick in collaboration with Raz Almani, who is here at the conference. The GPCR work was carried out almost exclusively by Evan Van Alst. I would like to thank all of my undergrads, my collaborators. A lot of this data for the cholesterol dimer structure was acquired with the help of Chad Rainsford and Amar Pham, who was really solid in helping us out when we were closed down and he wasn't. And some of our data is on his user group poster, and I'd like to thank you for your attention.
Thanks, Ben. So questions for Ben?
Hello.
Great talk. I'm so sorry.
You are first. You can.
Great talk. I just had a quick series of questions. From what I understood is that the KIR channel requires anionic lipids for activation, but then you also see cholesterol interaction. Is it at the same site?
It's almost the same site. That's the whole. So cholesterol and anionic lipids, though those binding sites are very close, which is why the interaction is competitive. And what's interesting is in like the classical KIR channels, which is what Kerbach is related to, it's completely competitive. If you go to something like GERK, which is something we also study, we have, we're making the eukaryotic channels now. In GERK channels, the action is actually cooperative. So actually cholesterol and PIP2 in that case work together. So there seems to be a lot of diversity among these channels for how they, but the binding pockets are all close to each other.
But then is cholesterol also able to activate the channel?
Not Kerbach. It's able to help activate GERKs, but not classical channels.
Okay. And do you see an effect with other anionic lipids like cardiolipin or other negatively charged lipids?
Okay, so for Kerbach, Kerbach will be activated by PGPA and cardiolipin. Now, classical channels are going to be activated almost exclusively by PIP2. Okay, now Kerbach actually binds PIP2, but it's PIP2 is slightly too big to open it all the way. Which is kind of one of the weird things about it.
Thank you.
Yep.
Nice talks, thank you. I was going to ask if you use cholesterol dimer as a rigid body in your Explorer calculations.
I'm sorry, what?
I was going to ask if you use cholesterol dimer as a rigid body in your Explorer calculations.
Oh, yeah. Oh, well, no, okay. For the first round, yes. At the final step, we allowed them to actually, we allowed everything to move. So, but for the first, so when we first docked it, the first step of annealing, we used it as a rigid body, but then once we had it in there, we allowed everything to have some freedom.
And it relaxed a bit, right, between two modes?
Yeah, it stayed together. Yeah, I mean, so if those distances between the cholesterols are really tight and they're unambiguous. So for the protein, we had like, over 50 distances, but about half of them were ambiguous. So we had to use ambiguous peak assignments in Explorer, whereas for cholesterol, it was completely unambiguous. And so the dimer did not come apart.
Thank you.
Yep.
Another question.
Yeah, so with regards to the COMPASS scoring,
Mhm.
it was a little unclear to me. So because COMPASS doesn't take into account spectral intensity, and it's
Yep.
only for a carbon-carbon spectrum.
Mhm.
And it was a bit unclear to me how far that got you with regards to your resonance assignments.
So in that
Or
particular,
structures.
so we are in the process of working that out, but for like C alpha C beta identification, it appears that it's actually pretty helpful, right? So basically what you can do is like in the current iteration of COMPASS, you wouldn't want to trust COMPASS for nitrogen shifts anyway, right? As we actually had discussions earlier today, N15 chemical shift prediction is not always that great, but for like looking at like consecutive C alpha C betas, from what we can tell, it actually seems to be pretty useful to help you get entry points. Now, you of course then still have to look for what would be the connecting N15 shift, but you have a pretty good idea of that based upon where those like, candidates are going to be in the protein.
And so with regards to which experiments you still have to perform, do you still
Oh no,
carry out your-
we're doing the NCACX, NCOCX, CA, NCO, CACO, yeah, all of it.
Okay.
So no, you still have to take all the data, but it's just an extra layer to help you with like assigning it more quickly, but then also just basically trying to check for self-consistency.
Great, thank you.
Yep.
Okay, any further questions? Then please join me in thanking all the speakers from a great session today.